Okay. My name is Niki Sporrong, and I am responsible for the eel issue at the Fisheries Secretariat in Stockholm, which is an NGO secretariat specialized on sustainable fisheries and ecosystem-based management. And we wanted to organize this webinar today in light of the ICES advice last year, obviously recommending a complete closure of all eel fisheries, and the ongoing Commission consultation on further measures to aid the recovery of European eel. And one very important aspect of this is the ongoing restocking efforts and how we should handle this in the future. So this webinar is intended to shed some light on different aspects of eel ecology and restockings, and look at how it's been done, the risks that are associated, whether it is an effective conservation measure or not. I don't know, Lina, do you want to just say something a little bit shortly about the practicalities of today's meeting, how things are going to work before we introduce the first speakers?
Sure. Yes, my name is Lina. I work at FishSec as well, and I just have some practicalities for you attendees here. So during the webinar, all attendants would be muted and not visible. It's just us moderators and the panelists will be visible and able to speak. But you can use the Q&A section to ask your questions, and that's found at the bottom of the screen. And we will try to answer as many questions as possible after each presentation. Also at the end, there will be a session with some questions from the audience. Please direct your questions to the speaker that you want to ask the question from. And if you have any technical difficulties, you can also let us know that in the Q&A section. And lastly, we'll record this and put that on the FishSec YouTube channel after the webinar, in case you want to look back at it. Okay, so back to you, Niki.
Thank you. And so do we also have Jan-Dag with us now?
Yes.
Oh, excellent. Great. Okay.
Hello.
So the first presentation will be from the Baltic region, but a wider perspective as well, and will be shared between Professor Dr. Reinhold Hanel, who's the director of the Institute of Fisheries Ecology at the Haenen Institute, and has also done quite a lot of his own research on eel, and Jan-Dag Pohlmann, who's also at the Haenen Institute, but also chair of the ICES IFAC GFCM working group on eels. I don't know if I did that in the right order, but anyway, both of you, long-standing experience of work on eel, different aspects, and good overview. So I leave the floor to both of you. I think Reinhold is starting.
Yes, thank you very much, Niki. I hope you can hear me well, and you also see my presentation. I want to thank the organizers for this workshop. I think it's a very timely workshop. There is a lot of discussion at the moment at various levels on the future of eel management. And therefore, since eel restocking is a widespread measure for the management of the European eel, I think it's worthwhile to talk about it and discuss the pros and cons of the restocking of eels. Well, but before I, let me see how I get this. Yeah, so before I get to the eel itself, I really wanted to make some general considerations about restocking. I think restocking is a very widespread measure for the management of fish. There is different reasons why people would consider to restock fish to natural habitats. And this has to do on the one hand with degraded wild stocks caused by habitat change, over-exploitation, but also the need to enhance fish yield where production is low. I think all these measures are valid, and they basically come, or these ideas, the idea to restock out of these ideas is valid, and most of these ideas actually come initially, I think, from salmonid restocking or other freshwater fish restocking. Nowadays, I would say that even in salmonids, or especially in salmonids, many of these measures are also heavily criticized. But still, there is the need for restocking for these causes is seen by many people. So overall, there is always the question also about the motivation of stocking in a way. Why do you want to do that? And I think one issue, and the picture that we see here represents that very well. Everybody is happy restocking fish. So whoever restocked fish, brought fish from captivity to the wild, had some kind of a very positive feeling. I can assure you, I did that in my past, working in a hatchery, and one of the best things ever done was to restock fish, because you always have the feeling that you're doing something good. On the other hand, restocking often aims at increasing financial return. And this is something I think everybody has to agree. A lot of money is earned on restocking purposes. And very often, is it to increase stocks for commercial fishing, or is it also the production and also the sale of fish for restocking brings some financial return, which sometimes can be significant. But one important thing to consider is that stocking of natural habitats, of course, is usually a significant human interaction with an ecosystem. And I think this is also something, there is no doubt on this, that bringing in fish to a natural habitat, some of them were either artificially grown or at least artificially cultured or kept in captivity for some time, will have an effect on the habitat itself. So even FAO has kind of created some objectives of stocking, or has argued about some objectives of stocking. So you can do stocking for mitigation, especially of disturbances to the environment, mostly caused by human activities. So to compensate for these human activities, stocking is one measure of mitigation. Stocking can be for stock enhancement, especially to maintain the productivity of a water body. If, for example, fisheries is high, stocking can be used to increase the level of fisheries in some freshwater, also in marine habitats. This is done in some areas. Of course, also to compensate for recruitment overfishing. Stocking can also be done for restoration, to restore fish populations, to retain stocks of threatened species, or to create even new fisheries. So sometimes new fish species or already extinct fish species are stocked to create new stocks that are eventually also targets for fisheries. But, and this is, I think, more and more obvious from various scientific investigations, there is a lot of risks with relation or with regard to restocking. Of course, some are mentioned here. Stocking increases the risk for increased intra and interspecific competition between stocked and wild fish. Another very important issue that also has been picked up by ICES in several occasions was that restocking, especially of capture of captive reared fish, can lead to selection and genetic drift in captive versus wild populations. There is also more and more knowledge about genetic changes in captive reared fish, so epigenetic changes that distinguish stocked fish from wild fish. There is, of course, always a risk to biosecurity and the spread of diseases, parasites, and non-native species. I think Olga is later on speaking about that. Of course, there is also increased predation on aquatic biota by stocked fish sometimes, and a shift in prey abundance depending on the quantity of fish stocked into native rivers. But, and I think this is one of the foremost critiques, is that restocking can be a distraction from other management decisions to enhance indigenous stocks. So in some instances, fish stocking actually leads to fewer activities with relation to fishing effort reductions or to restoration of habitat, simply because restocking is a more simple and sometimes also a more cheap action. And there are also, of course, many more. So this is just from a very brief look into literature, relatively new literature, being very critical towards restocking. Also, for example, in salmonid management, where I think the use of artificially reared fish for restocking to actually help the wild populations is heavily in debate. So how is it with the European eel? The European eel is, of course, unique in all this because of the very complex life cycle of the European eel, and also because of the fact that European eel is still relatively far from being artificially reared. So in comparison to salmonids and to most other fish species that are stocked, where the stocked individuals actually come from artificial reproduction, in eels, the stocked fish have to be captured as glass eels, mostly along the coast, sometimes even as yellow eels, and they are transferred and transported to other areas where they are basically stocked. So other than in other fish species, artificial rearing, artificial reproduction is not an issue. Juvenile eels have to be caught and taken from the wild to be transferred to other areas for restocking. So of course, the question is much more relevant even than in other fish stocks. Is restocking really a valuable measure to help the recovery of the European eel stock? And the rationale, of course, behind this restocking is that the eels must find in the recipient rivers where they are stocked, the conditions for the eels must be significantly better than in the areas where they are fished. And this is something which is scientifically heavily debated. Of course, the whole issue of restocking also drives the catch of glass eels as a source for restocking. One issue related to this is shown here, that of all these young eels that are captured every year, mostly for restocking, their fate is largely unknown. So just by maintaining this glass eel fishery, this also is one issue that the prerequisite that was brought up by the eel regulation, that at least 60% of these juvenile eels should be used for restocking. The latest numbers that were shown by the ICWG eel working group show that almost 60% of these juvenile eels, their fate is unknown. So only a relatively small fraction of 12% was used for restocking in 2011. So what are the rationales that you hear all the time when it comes to restocking? I mean, we debated this very much in the past months, especially when the European Commission asked for the regions for more input towards a better protection of the European eel. A lot of discussions also were held on the rationales behind restocking. And some of the most important arguments I want to repeat here. The first one is that there is still a surplus of arriving glass eels in the donor areas that needs to be harvested. This is something that is brought up very often, but effectively there is no real scientific basis for this statement. In only a very few cases, people even tried to find out what could a surplus be for arriving glass eels in certain estuaries. And I think this will be mentioned later on in other presentations, that there is no real scientific basis for this statement. Another rationale or another statement being made very often is the glass eel catches for restocking purposes help avoid density-dependent mortalities in the donor areas. This goes into the same direction, and it basically says if you don't catch glass eels where they arrive in a certain quantity, then density-dependent processes will lead to higher mortalities there. Again, something that has never been scientifically proven. It would be very interesting to know what really happens if glass eel arrive at an area where the density is already high. Does that mean that they will be faced higher mortalities, or is it also possible that these glass eels will rather stay in estuarine environments or in saline environments? There are, for example, there are reports that even in saline environments, like in the German Bight, during high numbers of glass eel arrival, there was a standing stock of yellow eels that disappeared completely in the recent past. So it is more than unproven. It's really completely unknown if there are such density-dependent mortalities in the donor areas that would justify glass eel fisheries. Another thing very often heard, that restocking has overall a positive net effect for the European eel. And this is also something which is very much in debate. What you have to keep in mind is that at least the catch mortalities for glass eel catches is now at about 10%. Well, there are different literature citations on these. Older reports that probably are outdated now speak from about 40% mortality in glass eel catches. I think everybody hopes that this has been significantly reduced. Newer numbers talk about something between 7 and 10%. But you also have to keep in mind that there are transport mortalities and there are also mortalities during restocking. And this all means that this has to be compensated in these rivers where eels are restocked. So that means that the living conditions, the survival rates have to be at least 20% or even higher than in the donor rivers. Otherwise, the whole concept of restocking of eels wouldn't make any sense. Another issue that is very important, especially for the northern countries, that a lot of eels for restocking are actually on-grown before, and they are on-grown very often in commercial aquaculture. And this means that they spend quite a long time under rearing conditions in commercial aquaculture with all the potentially negative effects that I wanted to point out before with regards to salmonids. So the only argument that remains basically is that restocking increases local stocks and helps, for example, in achieving biomass escapement targets like the 40% targets of the European eel regulation. And I think this is an argument that is valid. The more eels you drop into a river, the more eels will be in this river. This is very trivial, and but it's clear. Being in that river does not necessarily mean that these eels will be able to make the way back to their spawning grounds as silver eels. So it just means the more eels you drop somewhere, the more eels will be there for some time. And based on this single scientifically proven argument, restocking is still a measure that is of high importance, for example, for the achievement of the European eel regulation. So, but what does the European eel regulation, and especially the 40% target, do with this in return? The problem is that being such an important measure in many countries for the achievement of the 40% escapement target, this means basically that by using restocking as a measure, this introduces a legal obligation almost for restocking in areas of low or absent natural recruitment. Meaning that to fulfill the 40% targets in areas where there is no or almost no recruitment, managers are actually forced to restock in these areas to fulfill their management target. Another issue is that this enforcement, this legal obligation also drives at least in part, the high prices for glass eels on the market. And one issue about this is that most of these costs are actually covered by taxpayers via funding from EMF and MFAF. Another issue is that restocking is not limited to useful or to suitable rivers or freshwater bodies, but very often and to high quantities, juvenile eels are restocked upstream of hydropower plants and other unpassable river obstacles, which actually drives turbine mortality in these river stretches. And on the other hand, at last, these restocking purposes or these restocking actions actually sustain a completely artificial yellow and silver eel fishery in areas where there is almost no or very reduced natural recruitment. And I think this is really a critical thing, because restocking is very often sold as a measure for the recovery of the stock, but it actually sustains the fishery on eels, which in return again increases anthropogenic mortality significantly. So thanks for your attention. I stop here and give the word to Jan-Dag Pohlmann, who will for sure pick up some of these issues in a more detailed kind of way. Thank you very much.
Just going to come in here, Jan-Dag, while you're setting up, to say that we're a little bit behind schedule. We'll see how we can manage that further along. But welcome to provide your part of the presentation, Jan-Dag.
Yes. I assume you can see the presentation?
Yeah.
Then I will rush through it to save us some time.
Yeah, don't rush too much. We still want to hear what you're saying.
Yes, I will just show some of the data and the science that we have on glacial fisheries and restocking. And obviously, the first question, sorry, yeah, now it works, is where's the stocking material coming from? And as you can see here, I'm sorry for the small labels. This is the landings in tons by country. And then the point here is really it's mostly coming from one country, and it won't be a surprise to most of you that this country is France, with about 50 tons of glacial landings in 2019, making up for more than 80% of all the glacial landings. Within France, it's mostly the Bay of Biscay where the landings are coming from. So this will be the focus when we're looking at donor water bodies. So the important question is there a surplus, or are there density-dependent mortalities, as we've heard before? And you've all seen this graph of recruitment before. It has dropped significantly, and the red line would be the one that represents the drop in recruitment. And the elsewhere series, and France is largely part of that one. And we are at about 7% recruitment. So it looks a lot like there's not much eel coming to France. So how can there be a surplus production? It is not that easy, however. There are studies. To cite one is ACU from 2011 in the Frimur catchment. They've looked at the eel densities and whether there is a density-dependent mortality or surplus production. And they actually concluded that this catchment is saturated. There is varying recruitment, but at a certain later stage, the number of eels remains rather constant. And there are some other facts, I won't go into detail here, that tell us that there's, with a high probability, there's some sort of saturation mechanism. However, the Frimur is a very small catchment in the northwest of France, and it's doubtable that these findings can be, well, the same findings will account for the larger river systems like the Loire or the Gironde, which are quite important in glacial fisheries. Another point that's made in this study is also, and we heard about avoiding other measures, that habitat restoration might circumvent the problem, because the system might be saturated because a lot of the habitat has been destroyed. And I'm not talking about barriers. It's mostly about growth habitats for young eels, especially in the upstream river sections. So there are indications, but it's not everything there is. If we're looking on a higher level at the whole of France, and we look at the landings of yellow eels, and also up here this is just after 2000, we don't have longer time series, but the abundance of yellow eels, this has been dropping and largely paralleling the drop in recruitment. And that doesn't exactly say there is a saturation with glass eels in these areas. So it's not black and white, but we are far from saying the catchments in France are all saturated with glass eels. That's the important point, I guess. Now, where are the eels stocked? This is more diverse than where they are caught, but as you can see, on-grown eels are almost exclusively stocked in one country, which is Germany, with 50 tons in 2019, but also Poland and Denmark. Glass eels are stocked in more countries, but again, Germany stocks the majority of glass eels, and then there's France, Sweden, Netherlands, etc. In a nutshell, eels are mostly being stocked in Central Europe and in the Baltic region. And in this region, and that's quite interesting, I think, we have quite active fisheries. These are the 7 countries with the most stocking, and these are the landings, fisheries landings in this area. And these 7 countries make up for roughly 60% of all the landings of yellow and silver eels that we have in Europe that were reported to ICES. So there is quite an active fishery. It's not just about fisheries. I would have liked to show the same graph for hydropower, but we don't have the data. What I can show you is sum A, which is the sum of anthropogenic mortalities, which also tells you more. You can have high landings because you have a large stock and a low fishing mortality, and the actual mortality is interesting. There's a threshold that's out there, 0.92. At 0.92, you would achieve 40% of the production that you would have without anthropogenic mortalities, also meaning 60% of the production are lost to mortalities. And not too many countries are above this threshold, but it is quite difficult to calculate, and it's quite complex, and these data are far from being comparable. The point is there are quite significant mortalities in the countries where eels are being stocked, but they are hard to quantify. If we're looking at a map of Europe, this is from the European Environment Agency. On the left-hand side, you see the impact of barriers, and whenever you see something pink or red, this is a significant pressure from barriers. I won't go into the definition, but as you can see, also in Central Europe, particularly in Germany, there's high pressures from barriers. And this is not to say you can't stock there by any means. This is just to give you an idea of how many barriers exist and how complex the issue is. And Central Europe and the Baltic region are probably not the ideal spot. And the same accounts for chemical pollution, an aspect that is often not mentioned. In the donor area here in France, we have a light blue, which means a better chemical condition of the water bodies. And again, in Central Europe and in Germany, the chemical status is not that good. So there's also some doubts whether it is a measure of recovery if we're transporting eels from good areas to not so good areas. Then we have obviously some other risks. We already heard it's wild-caught eels, so the stocking material is less controlled. It's a vector for pathogens that has been shown for herpes virus, that has been shown for Angelicola. So this is quite obvious. We've heard about the glass eel fishing mortality. There's a regional adaptation of eels when they arrive. At least that has been shown. I know of one study from Cote on American eel, where they raised eels from glass eels from two different origins under similar conditions, and they showed differences in growth. So there might be other regional adaptations. And if we move an eel from one location to another, it might just not be adapted, or it might be performing better than natural recruits, and we don't really know what we're messing with. Same accounts for sex ratios, which is sex determination is density dependent in eels. So if we change densities, we also alter sex ratios potentially, and this might cause issues. We will hear a presentation on the migration potential of stocked eels, I guess, later, so I won't go into detail. The same on local ecosystem effects. I'm very keen on hearing these presentations, quite interesting. But there's a lot of risks involved in restocking. That's the bottom line. And this sort of had to lead to the current IC's advice, because in a nutshell, what we can say, we don't know if there's a net benefit of restocking. There's a good chance there is no net benefit. We don't have all the data on carrying capacity, mortality, and performance of stock deals. And at the same time, there's risks involved that we do know of. And if you're following the precautionary approach, well, then you won't do something where you don't know if it's good, but you know there's probably something going on that might be a risk. And one very important point that's not really spelled out in the IC's advice, but it's there. Escapement doesn't equal spawning stock biomass. So even if fish are stocked in a local EMU and escapement is increased, we don't know if that actually will contribute to the spawning stock. If you stock an eel in the Baltic, the chances that it reaches the Sargasso Sea might be much smaller than the fish that escapes from France. So escapement is not spawning stock, and we don't know the effect of stocking on the actual reproductive potential of the stock. We don't know the spatial contributions of different areas to the spawning stock in the Sargasso Sea. And then following the precautionary approach, the idea is not to say, well, glassy fisheries and stocking is allowed and we shall see where it should be forbidden. The precautionary approach dictates, well, there shouldn't be any glassy fisheries and there shouldn't be any restocking according to this, unless, and that is also in the advice, there is a proven conservational purpose. So if there's proof that mortalities are lowered, and it's tricky for the reproductive potential, I have to admit, but if there's, let's say, a high probability that you reduce mortalities and that you add to the reproductive potential, the advice shouldn't apply. That's particularly stated. That's explicitly stated in the advice, and this is something that hasn't gotten that much attention, I guess. And yeah, I rushed through it. Hopefully, I haven't watched what time it is, but that's it. Thank you.
Thank you, Jan-Dag. I think what we're going to have to do, so Argyrios, I hope you're ready to go. And we did start a bit late because we're waiting for all the participants to come in. So we might just take one. I don't know what we're going to do with questions. Questions might have to wait basically till the end until we've caught up with time. I think that might be an emergency measure here. Anyway, I would like to introduce Argyrios Sapounidis from the Fisheries Research Institute in Greece. He is also part of the IFAC GFCM working group on eels, and he has been part of the recent GFCM science program on eel to collect all of the available data and pull it together in the Mediterranean for the more permanent measures that GFCM is going to agree on to manage eels over the coming years. And there's some very interesting results about this presented earlier this spring, and I hope Argyrios will share some of them specific to restocking today. Welcome, Argyrios.
Thank you very much, Niki. Thank you very much for the opportunity to present you some of the results that we came up through this GFCM project, and it's quite interesting. So I think that you can see also, everybody can see my screen now. So let's begin with a short description of the project. It started almost 2 years ago, and the main goal of the project was to find and record all the available data that exist in the Mediterranean countries regarding eel fisheries, recruitment, management plans, local plans, etc. And to tell the truth, we had some very, very interesting results. And here we will see some of them regarding the management measures that established in the countries. So, just to begin, since 2008, European Eel was classified as critically endangered, and EU regulation 1100 in 2007 proposed to the EU countries to establish measures for the protection and the recovery of the stock. So the main targets of the national management plans was to reduce the anthropogenic mortality and at the same time to increase the escapement of silver seal during the export migration. One of the measures that almost all countries have established was the restocking actions, but we found that only Italy and Greece are performing actions like this, in different degree, of course, but they do until today. In the Mediterranean, the prevailing eel habitat is the lagoons. You can see here that almost 760,000 hectares are currently available to eel, and the fisheries are taking place in lagoons. Out of the 700 sites that during the project were identified as fisheries grounds, 276 corresponds to lagoons, and also the almost 40 or more than 50% of the catches are coming from the lagoons. The lagoons fisheries is a specific species-specific fisheries because they're targeting mostly Eurhaline fish that migrates towards the sea for migration or for spawning. And this is the way that also eels are fished over there. The traditional lagoon management was frequently used for restocking as a tool to enhance the lagoon yields. They collect the catch fin fish fry and they transfer them in the lagoons to sustain the fisheries. This is a practice that started in the 19th century and still remains as an active practice, mostly in Italy, in the North Adriatic area. And this practice was then adopted by the fishermen in other Mediterranean countries. But this kind of practice is overlapping with the fish farm type, and that's why it's called also as an extensive aquaculture. The Italian method of collecting fish is using specific gear like eel fight nets or small sea nets to collect fry, mostly for eels, glass eels, mullets, sea bass, and sea bream, and then introduce them in the lagoons. Until the 70s, this fisheries was not organized, but most of the glass eels that were fished for this purpose was used also for direct consumption, and there was also a high rate of poaching. During this, for this practice, specialized fishermen were authorized to move along the coast and collect and fish glass eels, which were transferred with trucks equipped with storage, specific storage, and sold for aquaculture for extensive and intensive. And this practice was continued until the 80s and declined in the 90s along with the decline of the stock. Following the regulation, the Italian management plan foresaw a number of measures to be implemented in the short, medium, and long term. As such, there was a total closure of fisheries, both commercial and recreational, and in 11 regions out of 20, maintained a commercial and recreational fisheries, but by reducing the fishing effort in 9, restocking was implemented in 7, silver eel releases in 2, and habitat-related measures were established in 9 of them. For the glass eel fisheries, there was a new legislation that was established in 2 EMUs in Tyrrhenian coast, where specific regulations were established for this purpose. Glass eels, you can see here in this table that the restocking that was implemented during the period 2011 to 2020, it happened in not all the EMUs, and they used wild glass eels, but also on-grown eels that was imported from other countries, mostly imported from France. The main problem in Italy is that there isn't any experience, or it was not well documented regarding the impact of restocking in the local population, positive or negative. And in some cases, restocking was implemented in sites with modalities that actually aimed at enhancing the escapement, but also there were some restocking in areas that the effectiveness of these measures is in doubt, mostly because of unsuitability of the seed or unsuitability of the sites. Reduced quantities, of course, because of the decline of the stock, and also now because of the increased interest in restocking has led to the reassumption of glass eel fisheries, increasing also the fish-seeking effort, and stimulating also the illegal glass eel fisheries. Also, the cost is getting higher and higher, and there isn't any specific or an appropriate reporting system. On the other hand, in Greece, based again on this EU regulation, there isn't any recreational fisheries. It's completely closed since 2009. Fishing of eels also, eels is allowed to be fished only if they are above more than 30 centimeters in total length. And in this way, glass eel fishery is completely prohibited, and yellow eels are partially closed, and only, as I said before, only those eels that are above 30 centimeters are allowed to be taken away. The Hellenic Eel Management Plan foresees also that in case of restocking actions are taking place, special permissions can be issued for glass eel fisheries. But until now, there wasn't any request for this for such actions. However, the management plan considers a release of 10% of the imported glass eels that are used for rearing purposes, and also 30% of the annual silver eel landings in each lagoon has to be released to the sea in order to achieve the 40% of escapement. However, there isn't any evaluation system. We are not sure of the effectiveness of these restocking actions, and we are not. And sometimes we are not sure that the 30% of silver eels are really released in the sea. Until 2017, there were 4 fish farms for rearing eels in the western Greece. After the Europol seized in 2017, only 2 farms are still active, which are importing glass eels from UK and France. Based on their imports, as we mentioned before, 10% has to be released. So this so all these years from 2013 until 2019, the glass eels that were imported for rearing was from 155 kilos up to 1,598 kilos. And 10% of these were released, as you can see in the graph. And you can see also the 30% of silver eels that are released back to the sea during the sporting migration. During the GFCM Eel project, we decided also to do some evaluation of the possible measures that can be established and towards this effort for the recovery of the stock of the population. One of the measures, one of the scenarios that we examined was also the restocking in all sites in the Mediterranean using 1 gram of glass eels per hectare. So this scenario was completely theoretical, and we take into consideration the average of glass eels landings in the area between 2015 and 2020, and equally distributed and released in all sites. So we are talking for 1,061,000 hectares. And we take also into consideration economic and biological consequences. And both landings and escapement increase even if at low levels, and the effects are evident where restocking densities are closer to the actual recruitment. So here you can see the results of these scenarios. You can see here in the restocking in all sites with 1 gram that there was 0.3% increase in the landings, but also had a 3.2% of increase of the silver escapement. But it's not the best case scenario. You can see that we can achieve even higher percentages of silver eel escapement just by implementing measures for fishery closure or reduction of fishing effort. From the Italian experience, we can see that with glass eel fisheries, we interfere with the national recruitment dynamics. Restocking actions do not substantially enhance natural recruitment, but we have a local enhancement which not guarantees the improvement of the survival of the species. We still don't have any signs of recovery, even if there are eel restocking actions. We maintain the fishing effort on this sensitive juvenile state, which otherwise it has to be protected. There isn't any evidence of the effectiveness of this measure, since we don't know the percentage of the mortality, if it's increasing or decreasing. And we mostly rely on glass eels that is imported from other countries, mainly UK or France. So we have mortalities in different stages of the transportation of the glass eels of the seed. So during the transportation, the quarantine period, and during the release of the glass eels. And the use of the establishment of measures of restocking diverts the efforts of implementing other more effective measures, such as the decrease of fishing effort or completely closure of fisheries in the areas, in the fishing in the areas in its country in the Mediterranean. So the benefits of restocking are less of the risk that we are taking for restocking. So our perspective through this project is to agree with the ICES advice that glass eel fisheries should be reduced or even closed, and restocking actions are not that effective towards our common goal, that is to the recovery of the stock and the protection of the species. And that's all from me. Thank you very much.
Thank you, Argyrios. And yes, so we're a little bit further behind now. We're just simply going to skip the question sessions after the presentations. I hope we will have time for some questions at the end after the panel. And otherwise, Fisheries Secretariat, we'll capture the questions and maybe discuss whether we could instead have some of the questions answered after the meeting and circulate to the participants. So I hope this is not going to be too unsatisfactory for participants, but I think the most important thing is to listen to what our invited speakers are saying today. So I would like to move straight on to Caroline. Yes. So Caroline Durif is based in Bergen, Norway, at the Institute of Marine Research, and she has a long-standing experience of research on eel. She's specifically focused on the ecology of eel, life history of eels, and eel migration. And she's going to tell us a little bit about one of the latest studies she's done on eel migration globally, I should say, not just the European eel, but other species as well. The floor is yours, Caroline.
Yes, thank you, Niki. So I'm going to take you through the fascinating story of the magnetic orientation of eels, and more specifically, the cues, the orientation mechanisms that we think that eels use when they migrate from their growth area to their spawning area in the Sargasso Sea. So we understand how larvae drifting, hatching in the Sargasso Sea and then drifting with the Gulf Stream are dispersed throughout Europe and Northern Africa and way into the Mediterranean. They drift with the Gulf Stream. But it's a much bigger challenge to understand how all of these eels from such a broad geographical range manage to navigate to the Sargasso Sea to meet all at the same time at the same location. Because there are no obvious cues in the open ocean, except for one, which is the Earth's magnetic field, which has the advantage of it's present day and night, and it's unaffected by weather. So it can be used everywhere on the globe, and it varies in a predictable manner. So we also use it as humans, and you can derive three parameters from the Earth's magnetic field. And the first one is direction, which is your usual compass. So that's where the arrow points to, magnetic north. And the other parameters are derived from the intensities. The intensity of the field, as you see, there's lines around the Earth, and so there it varies so that its lowest, the intensity and inclination, that's the angle between the magnetic lines and the Earth's surface. These are lowest around the equator and they increase towards the pole. So basically, if you're able to sense the intensity and the inclination of the field, you can figure out where you are on a latitudinal gradient. So there's many animals, many species that can do this, starting from magnetotactic bacteria, which have magnetite inside them, to higher level organisms, such as mammals. It's used for orientation, but also for other physiological cues. So here in Norway, we have a very special, unique lab, which is located in the middle of nowhere because we don't want any electric disturbance. We're working with very low intensity magnetic fields, and it consists of a big coil system. That's how you create magnetic fields artificially. You just loop electrical current, and if you change the direction of the electrical current, you change the direction of the magnetic field inside the loop. And if you increase or decrease the intensity of the current, the electrical current, then you increase or decrease the intensity of the magnetic field. So we can play around, simulate magnetic fields, change magnetic north. So the first set of experiments we performed, which was on yellow and silver eels, And so basically, the principle is that we shift magnetic north. So there's all other cues are hidden. There's no external cues. It's inside a building. It's in the dark, and all we do is shift magnetic north, and then we look to see a specific behavior of eel if they also change their orientation. So that's what we found. We found that yes, they do change their orientation according to the magnetic field, and that's also temperature dependent. So basically, when they're in a migratory mood, they orient towards the flow, and otherwise they don't. They display a sort of we call it a bimodal orientation, which would be a shoreward orientation. The next set of experiments was we did with glass eels, and that was mostly the work of our former PhD student, Alessandro Cresci. And we used this device, which was developed by Claire Paris at the University of Miami. And here's a short video to show you that's, so we deploy it. It's a drifting aquarium. We deployed from small boats. We put the fish inside the chamber, which is made of a plankton net, and then we film it. Yes, and this is what we see. This is a time lapse from inside under the chamber. And so we have compass on the system, compasses, so that tell us, so that the device drifts with the current. So the larva or the juvenile, the glass eel, just follows the flow and can orient freely. And then we film it. And then we use these videos after in the lab, and then we track the paths, and we can retrieve the swimming speeds or the orientation. So what we found is that whether the glass eel were in the mag lab or in the in situ, in the drifting chamber, they showed the same compass orientation. And so again, in the magnetic lab, there's no external cues. So the only thing they can use is the magnetic field. And also this orientation changed according to the ebb and the flow. And this is what they do in nature. They just orient differently according to the tide. So we also looked at, we did further experiments, and we saw that what they do is we collected glass eels from different streams, and we saw that the orientation that they register is different according to where we caught them. So they imprint the estuary for where they tried to go in. So at least for a few hours. So, or even days. So we know now that eels have a sense of direction, the compass orientation, magnetic compass orientation. But now the question is, can they sense the intensity or the inclination of the field, which they would require to navigate to the Sargasso Sea. So how do we do this? Well, this summarizes the magnetic map hypothesis, which is basically that the larvae hatching in the Sargasso Sea would register the geomagnetic parameters, so that's the intensity or inclination of the magnetic field, and maybe also the gradient as they drift with the Gulf Stream. And the question is whether they can retrace this magnetic route. So to do this, we calculated the magnetic intensities that the larvae experience, and also that the adults would experience or try to follow during their spawning migration. And so we did this for the European eel, but we also did this for the sister species because they all have a common ancestor. And if this is a mechanism that works, it's probably the same in the other species because they all have distant, mysterious spawning areas. And so we chose the species for which we have some data about where the larvae are or the tracks of the adults, and in the northern hemisphere, but also in the southern hemisphere. And this is what we found. So basically, as you can see, so these are the values of the magnetic intensities, and these are isolines, and they're parallel to the equator. And yes, they increase towards the north, obviously. So the larvae of the American eel and the European eel, during their drift, they always experience increasing magnetic intensities. And the adults, it's the opposite. If they retrace, if they go down, decreasing magnetic intensities, they would end up in the spawning area. And interestingly, we found the same for the Japanese eel, even though the magnetic configuration is slightly different, but given the currents and their drift, it's the same. They go down, they experience increasing magnetic intensities. And so do the Australian and New Zealand eels in the Southern Hemisphere, which is interesting because it's an opposite configuration. And that is because the spawning area is located north of the growth area. So it fits. And then the adults, the same. If they go down decreasing magnetic intensities, that's towards the blue, then they would reach the spawning area. So to find some evidence of this, we looked at data from other scientists that have been tracking and tagging eels using pop-up satellite tags and data storage tags. And these are from were summarized in David Wrighton's paper. And this shows the release points of some of these eels that were tracked. And as you can see also, the eel that was tracked the furthest was tracked until the Azores. And what it did is that it followed decreasing magnetic intensities. And then, although we don't have data after this, it's easy to see that if it stays on the isoline, then it'll reach the spawning area. Now, interestingly, the eels didn't take the shortest route, which you understand. If they follow the steepest decrease of intensities, that's what they would do. And the same for the American eel. It followed exactly a parallel track, going down, decreasing magnetic intensities. These are tracks from the Japanese eel. And here again, they didn't take the shortest route to the spawning area. But again, they seem to take this perpendicular track to the isoline, so the steepest gradient of magnetic intensities. And then it seems that if they hit the target isoline, then just following it, they would reach the spawning area. Now, this is a navigation tactic that is actually quite well known. And it's, for instance, if you aim for a target, you might miss it. If you aim directly for it, you might miss it. You might end up on the right or on the left. But if you aim off of your target and aim for a stream, like a line feature, such as the magnetic isoline, and then follow this line feature, then you won't miss your target. And this is especially useful if you're coming from many different locations and you all want to meet. And this is what happens with the eel. They don't really care about finding a specific location as long as they reach that isoline. And that's kind of the key of the model. So wherever, and this works for all of the species that we examined. And so the principle is that the larvae, they register that target isoline where they hatch, and then they drift along the whatever current, and then reach, live their life. And then later on, if they just follow the steepest gradient of decreasing magnetic intensities and reach that target isoline, the highway, and then just follow it, then they'll meet each other. And then the stop point might be simply because they've matured enough, they're ready, and then using pheromones, they might all congregate together. So what is the implication for restocking? Well, for instance, when eels are translocated from the Bay of Biscay to the Baltic, well, there's two different things. So eels our model works if eels imprint the hatching location only, but also eels might imprint the whole gradient, and that we don't know. But recent paper that shows some data of eels that were tagged and released in 2 different locations in Belgium and in Denmark. And even though they were released at the same location, they took different tracks to, these are silver eels. So some of them took the English Channel and some of them took the North Sea. And that makes me think that why would they do this is maybe because they imprinted different incoming ways at the larval stage or the glass eel stage. So it's likely that they imprint also the gradient, which would also help them come out of the Baltic. So also eels, when they, even if in the scenario, if we translocate them from the Bay of Biscay, some animals need to calibrate their magnetic compass. If they haven't experienced these high intensity magnetic fields, they might not be able to sense them. So this has been shown actually in other species. And one of the species which has a lot of magnetic orientation studies is the loggerhead turtle. And it's been shown, this is just an example, but when you simulate their position out of their usual migratory route, they display random orientation. They don't manage to orient if they're not in the known magnetic configuration. So we know that they have a compass, a magnetic compass, and they can register, they can imprint that direction, and they use that in continental waters. The magnetic map, that it's actually quite a simple mechanism, just based on magnetic gradient and on finding this target magnetic isoline. It's not very precise, but it works, and it cannot be very precise because there's the magnetic field changes over the years, there's solar storms, so there are some disturbances. So all they needed is a meeting point, and this is what we showed in our study. And but all in all, the evidence so far suggests that translocated eels would not find their way back to the Sargasso Sea. But again, that of course needs further confirmation, but this is what we have so far. Thank you, and if you want further information, and all of this has been published, so just go on our website, FishLarvae. Thank you.
Thank you so much, Carolyn. And obviously I should have said initially, but one of the most debated issues of restocking is the whole issue of whether restocked eels find their way back to the Sargasso Sea and actually contribute to recruitment and recovery of the eel stock. And for that particular reason, we thought that Carolyn's recent study was a very interesting piece of this puzzle. Anyway, we are still behind, but I think we do need a break. But we're just going to have a very short break. I hope people can be back for 1:15. So it's just a leg stretcher, really. And then we'll welcome Olga when we're all back. Thank you very much for now. Okay, everyone. That was a very short break, but I hope most of you are back with us. And Olga, are you about ready? I would like to introduce Olga Haenen. I hope I said that more or less correctly.
Yes.
She is the head of the National Reference Laboratory for Fish, Shellfish and Crustacean Disease in Lelystad in the Netherlands since 1985. She's a senior expert on diseases of edible insects for food and feed, and has just finished a 4-year professorship at the University of Applied Sciences at Den Bosch. But I know that you have also, very long-standing experience. I think almost 30 years have been looking at pathogens in European eel, which is obviously an important issue related to restocking. So I welcome you to provide your presentation, Olga. Very pleased to have you here.
Thank you very much, Niki, for this invitation. And I put now on full screen. I hope it works. I will talk on the major diseases and pathogens of European eel, and also a bit on the risks of restocking practices of eel, and potential effects on recruitment. And the latter subject I know the least of, and the first I know the most of. So you will see this during my presentation. Yes, so now I have a full screen. If it's also seen by you, I hope. I'm working at Wageningen Bioveterinary Research, which is actually the State Veterinary Institute of the Netherlands since 36.5 years. And here you see already a few pictures of healthy eel. The second picture is of eel with Vibrio vulnificus, a very nasty infection. Then you see a herpes virus, and you see the Anguliculoides crassus, which was my thesis subject. So enough on this. We have a lot to share, but I try to keep my 20 minutes. So the introduction I will start with, then I will go through the most important diseases and parasites of eel, parasites, viruses, bacteria, and then, of course, for today important, the interactions between wild and farmed eel, with focus on the risks of restocking eel and the effects on recruitment, with also some conclusions. And here already on the right, you see a picture of restocking eels, a nice picture made by Dirk-Jan Kraan. Well, I'm working at the shellfish disease laboratory. We are appointed by the government, by the Ministry of Fish, Agriculture and Fisheries and Food Safety. And we are National Reference Laboratory, as said. We have notifiable tasks for the EU and also for OIE kind of notified diseases. So we do diagnostics of notifiable diseases according to ISO 17025. It's the quality assurance accreditation of the testing. We have developed and implemented diagnostic tests and also maintain expertise on notifiable diseases. We give advice to competent authorities, and annually we do monitoring of mollusk disease twice a year in the Zeeland area, which is the southwest of the Netherlands. Apart from this, yeah?
Can I just break in? Because I think maybe your screen hasn't changed, and obviously I don't know if you want to change your slides.
Okay.
But I think that there is a risk that you are not sharing the full screen version that you're wanting to share.
Okay, so I will stop sharing and start sharing again, okay?
Okay.
Yeah. So what do you see now? Not yet?
No, mine is still the same as it was before. So basically shows your presentation with all of the slides on the left-hand side.
Okay, I'm sorry about that. Then I will, let me see, voorstelling weer eindigen. And then new share.
Now it's definitely moving.
Yes, so I will try again. And I will try full screen right now of the second slide. Perhaps you can see this. It's not yet. Is it full screen right now? No?
No.
No.
And it's you're on the introduction our laboratory. That's the slide you're on. I think it's the third slide.
Yeah.
Did you?
Well, then I will just do it like this, I think, and then I'll just make it bigger to that you can see it. I mean, otherwise it takes too much time. I don't understand why it's not really okay.
Okay.
So we now see if you are right, the introduction of our laboratory, right? Okay, thank you.
Yeah.
So we also do multidisciplinary diagnostics of fish, crustacean, and mollusk disease. So it means parasites, bacteria, viruses, and also food issues, histopathology, etc. And we give advice to international bodies and give sometimes fish disease courses. We work all in this new building, and our research interests are actually a lot on the eel. We are really the central eel laboratory of diseases in Europe, you could say, we've been working a lot on the herpes virus of eel, but also on Sapounid viruses, and the contact zoonotic bacteria of fish, of which Vibrio vulnificus is a very important one of eel. And then mollusk diseases, I'm not going to read this all out, it takes too much time, but also lately also a bit on the veterinary risk of edible insects for feed, for use in feed. So to use certain kind of substrates to culture insects and the risks of it, just experimentally. It's not yet used in practice. We have, so I said, we are the Central European Eel Disease Laboratory. People call us like that. We are not appointed like that, but it's because with my team, we had a lot of research and a lot of papers on this, more than 35 peer-reviewed papers on eel diseases. Please visit our website to have a look in this. Our clients on regarding eel are eel farms, eel fisheries. Also before restocking, eels are checked for certain diseases. Also the sport fisher boards, sometimes zoos, not very often, the government, of course, and the international and national colleague institutes. Multidisciplinary diagnosis already told you, and here you see an example, taking a blood sample from an eel, looking into Trichodina parasites, 2 different cultures of bacteria, which we then test with the MALDI-TOF. It's kind of identification method we have developed, and also have a kind of library database made on the fish pathogenic bacteria in this apparatus. And we did do a lot of cell culture. We can, but most of it nowadays is real-time qPCRs and a lot of histopathology for the shellfish especially. Also, I participated as one of the organizers of the ICES working group on monitoring of eel quality. So we have actually Claude Belpère of INBO, Belgium, and myself with the whole working group at Brussels. We have got together in January 2015 and developed a standard and harmonized protocol for the estimation of eel quality. And you can find the data via the ICES, as you know, probably. The Netherlands is really typical eel country. So we have important European eel producers. Actually, totally, we have a long tradition regarding smoked eel. And in the 80s, actually, the aquaculture, including eel farming, came up. And it also meant that we were ready for diagnostics. And the decline, well, this is, of course, a picture which everybody has different pictures for this, but it says what is causing the decline. Is it the fisheries? Is it the migration barriers? Is it pollution? Diseases? Well, I've marked it red, of course, because this is really this is about our work. This picture is actually on this one. This is our picture. And predation by birds might it be. But diseases, well, to my opinion, it's a kind of combination of all kinds of factors. And diseases might as well be a problem, because what is the basis for disease? When you have the eel and you have the sneezco, the 3 rings, you have the eel, you have the pathogen, and you have the environment. And where the pathogen is bad present and the environment is bad, you get the most problems, severe disease. When there is only a pathogen which may be quite nasty, then you already get light disease. And also when there is a water quality problem, without pathogen, you can also get problems. And it all depends, of course, on immune status, stress, etc., other factors. So there's a lot to do about that. Eel diseases in nature, you have actually a low infection pressure normally of bacteria and viruses. And mostly parasites are a bit more or less in balance with the eel. So, I mean, not in too large quantities, and so that the parasite survives and the eel survives, so that they can have benefits from each other. And before 1980, there were only a few papers on the eel disease in Europe. And since then, you see various remarkable eel diseases in literature. And why is this? Well, actually, I think because of aquaculture, there were more eels in focus for diagnosis. I mean, there were more captured eels, and when you see disease, you are going to start to research this and to investigate. So more knowledge on diagnosis came up from the 80s, more specific and sensitive diagnostic tests, also globalization. There was some import of live eel for eel farming from remote areas, and they also brought some pathogens into Europe. Not that we can fully prove, but it's not known that it came from elsewhere. So actually, first about examples from parasites. We have, like this one, Trichodyna. You find it everywhere, also in other fish species in the freshwater environment. And also the Pseudodactyl Argyrios, which is a kind of monogenic trematode worm. It's an egg-laying worm, and it has 4 eye spots, like you see here. This is a piece of gill. And these worms occur also in nature and in farms. And we have for instance, the white spot, also kind of ciliate parasite, the protozoan, nicely stained here, but it causes all kind of skin exudate white spots. So this is actually not fungus or whatever, it's just a parasite rolling in the skin and causing these kind of slime white spots. And now this is a real wild eel problem. This is Myxosporin parasite Myxidium giardi, which we just by incidence saw in the Lake IJsselmeer, together with Rivo, which is now Wageningen Marine Research. We actually discovered that these eels showed a kind of very regular pattern of all kind of small kind of, well, bulbs or granuloma or whatever you can call it, but it's exactly on the sideline. And when we kind of made slides of the sideline, histological slides, you see here that the sideline is totally filled up. And when we focused on this, we saw all these Myxidia here, which are these parasites filling up the sideline. And so these eels were totally disoriented. They were kind of moving very irregular, etc. So it actually, the balance was totally done, and they couldn't eat well, and they started to die. Just an example, other parasites you find a lot in the wild in blood, trypanosoma. It's a typical trypanosoma of fish, so not harmful to humans, but we actually investigated it, and we saw also in literature that Tesh in 1983 already said that there were actually parasitofauna, more than up to 44 species of eel parasites in the wild, and Kennedy in 2007 found 63 metazoan eel parasites in wild eel. And well, 3 introduced species are important pathogens for parasites, to say, not yet about viruses or bacteria. That's Pseudodactyl Argyrios anguillae and Abini. So these are the monogenic trematodes, the gill worms I showed you, and also the swim bladder nematode, the Anguliculoides crossus, which originated from Southeast Asia or East Asia, as far as we know. And I'm not gonna mention this all, but you see here examples of literature on these parasites, which is really interesting to look into, and to also see kind of investigations on these parasites. And this was my thesis subject. So I have also some pictures from our own work. From the 90s, 1995, I graduated for the PhD. I did this together with Van Banning from the Wageningen Marine Research. We actually, this is a healthy eel swim bladder with the rete mirabile to regulate the CO2 in the blood. So it's not having any parasites. And here you see 2 swim bladders with necrosis, severe necrosis, fibrosis. So kind of, yeah, thick layer caused by the parasites sucking blood from the veins in the swim bladder. It was originally described in Japan and in other countries in East Asia, and it came to Europe in around the time that I started my job, so it was really good timing. But of course, this was a very interesting subject to work on in the very start years of the parasite. from the mid-90s, like with every new parasite, you first have an acute phase, which was bad for the eel, for the wild eel, and for the eel culture. But then you saw milder lesions and lower number of parasites. So when you now look into the wild, you see a low number of parasites and a mild kind of infected swim bladder, which causes that the eel survives and the parasite survives. And here you see the female and the male depicted. So rapid spread all over Europe and beyond, from the Netherlands and Belgium. We were the first because we didn't need any sophisticated diagnostics to see the parasite. And some people even thought they were baby eels. You can imagine, but you shouldn't kind of restock them. Short life cycle via copepods, many species, and paratenic hosts, like also like sticklebacks and smelt, etc. There were many research groups working on this, and there are now even more papers than 120. There was a negative effect probably on the silver eels and the spawning. Swim bladder function decreased, swimming speed reduced, fitness and silvering affected, and stress tolerance reduced. It was, and I think it still is, a big question mark. Would these eels, these silver eels, then at the end be able to reach Sargasso Sea and spawn? We did a monitoring of wild silver eel to see the status of the silver eels. It's quite some time ago, so it was in 2005 with the LEUPF, Landes Ökologische Institute in Germany. And together we estimated eels. We took eels from several places, so it was a really nice joint project. Condition, necropsy, blood smear, parasites, bacteria and viruses. The River Rhine and the Lake IJsselmeer as well. We saw often, well, kind of hemorrhages, but these eels were very active, so that was not a real problem. What we did see is Anguliculoides in 50 to 90%, and one female may produce 4 million of larvae, just estimate. That's just the nature of the parasite. And we saw also in about 70% this trypanosome, not in high amounts, but well, it's in high amounts only. The eel is really getting kind of necrotic, how do you say? Well, kind of apathetic, etc., and less active. So we saw the aspecific hemorrhages in a low number of eels, and this is what we found. So they were all actually quite good condition. But we questioned, since we found so much also the unguided herpes virus 1, would it be also responsible for the decline of the wild eel? And this actually depicts the unguided herpes virus 1 infections we saw. And eel migration to the Sargasso Sea, stress, water temperature, sometimes ideal for unguided herpes virus 1, because this is a warm water virus. It needs temperatures around 25 degrees centigrade to replicate well. And then it depends really on the, well, the immune status of the silver eel, which has already made a long trip, if they can stand this. Eel viruses, to summarize, which we have been looking in, there must be more, but these are the most important ones. That's the herpes virus, the eel virus European, and that's the EVE. So that's actually a double-stranded RNA virus. And then the eel virus European X, that's a rhabdovirus. And rhabdovirus is the same group as the rabies viruses and a lot of other fish viruses. First, the unguided herpes virus 1. It's since 99, it's detected in the Netherlands since late last 89. And why? Because we imported the eel cell line from Japan. That was actually the reason that we detected it for the first time in Europe. And then we actually had the permission to distribute the cell line eel kidney 1 to different labs in Europe, and then it was detected all over Europe. So we cannot say that it started in the Netherlands. Sometimes you see no disease signs, but mostly you see kind of hemorrhagic pattern, also with the fins very red. And it is giving a mortality in the farms up to 50%. It's really stress related, like also herpes with humans and other animals. Optimum temperature 25 degrees centigrade. We actually first developed a conventional PCR, my colleague Rijsewijk, and then we had a PhD student, excellent, who did an excellent thesis on this virus. And also a lot of research has been done. Please contact me if you like to have the papers or look into our website of the Institute. FishSec's eel virus European is the second virus. It's in farmed and in wild European eel. And Van Ginneke et al. Already found it in the wild European eel. But this was actually a single finding. We didn't find it afterwards. We didn't test huge numbers of silver eel, because when we test silver eel, we have to kill the eel. And so we didn't have kind of monitoring or so, but sometimes we got silver eels for diagnosis. And mostly this is actually in combination with unweeded herpes virus eel found. So a double infection with eels, with viruses. There are all kind of publications on this, and I just go to the third virus, which is the eel virus European, the double-strained RNA virus. It's called bronchionephritis, so it's actually well, harming the gill area, and it's found in farmed eel, but also in elvers. It has been found since the 90s with a lower optimum temperature. It's closely related to the trout virus, IPN, which is found in Europe, all kind of places. And you see all kinds of spastic swimming, retraction of the abdomen, etc. So when there is a big stress factor, these eels will really die of this. And the mortality is can be very high, dependent on the factors. Then some bacterial diseases of eel, and of course this is not like giving a book in 20 minutes on eel diseases, but the most important I only show Pseudomonas anguliseptica in elvers from Southwest Europe. It was already found in the 70s in Japan, and since the 80s it has been found in Scotland, for instance, here by Tony Ellis and co-workers, who actually proved that you don't need any antibiotics against this bacterium. If you just heat the water to 26 degrees centigrade for 2 weeks, the elvers totally recover. And this is also used in the Dutch eel culture to, when Pseudomonas anguliseptica is diagnosed, they have a kind of whitish layer around them when the quality of the elvers is getting very low with all the transport. Etrachella tarda is a kind of bacterium which may be also for humans harmful, but in eel you see they can really, this is a wild eel, cause nasty infections. Fibrio angularum, it says already the name, this is a real eel bacterium, but it's also found in turbot and sole sometimes. Red eel pest and Fibrio vulnificus already mentioned. It's we did a lot of work and still do a lot of work on this also with the University of Valencia on fast diagnosis if we are having a zoonotic strain or just an eel pathogenic strain. So if the strain, the bacterial strain, is also harmful to, for instance, the eel farmer or the fisherman. Double infections occur also with bacterial virus combinations. And now I get actually to the interactions of wild and farmed eel. Well, to start with wild eel, we talk about open water instead of indoor recirculations or ponds. We start with glass eels in wild eel, but that's the same with farmed eel. With wild eel, we talk about seasonal temperatures, and farmed eel indoor, they have a constant temperature of, for instance, 25 degrees centigrade. Wild eel eat wild feed, and farmed eel eats fish eggs when they are elvers, and then they start to eat pelleted feed or paste. Low densities in wild and high densities in farmed eel, and wild eel have a natural water quality, whereas the farmed eel have artificial water quality. And here you see also a picture of farmed eel They all get closely together because they want to get some pelleted feed out of this feeder here above, which you just now don't see. But so that's why they are all together. The rest of the basin may be empty because they are just aiming to get the food. Well, the interactions of wild and farmed eel, risk of pathogen transfer. Yes, that's a question. When we start here, this is always the start. The start with elvers swimming up the rivers or arriving at the rivers. They had already a long trip and they had stress. And these elvers, in the wild, they would actually develop to yellow eel, and then they swim up the rivers, they become yellow eel, and they migrate, they become migrating silver eel and swim back to the sea. And of course, then I don't put sea and water and freshwater in this scheme, just wild eel and farmed eel, okay? But this is the freshwater phase, and then you get the, of course, the metamorphosis up to silver eel, and then the silver eel gets to the sea and swims back to the Sargasso Sea. Stress, long trip to Sargasso Sea. But if we go from elvers to farmed eel, so elvers already have some parasites, bacteria, and viruses in some cases. Well, not often. I mean, we've seen quite some portions of elvers, of which we got a few elvers to test and to sacrifice to know about the quality of the elver batch. When elvers swim up the rivers, they get pigmented, they may acquire Anguliculoides crassus. Just when they start eating, they get infected, they eat these copepods and get infected by this parasite. Okay, but the non-pigmented elvers, they are transferred then to the eel farm. Okay, here are the elvers. What is the quality of the elvers? They may already have kind of Pseudomonas angliseptica, like I said, when they have been too long in the lorries or whatever. It can be harmed, it can be cured without antibiotics. That's a good thing. At the farm, they are in high temperature. The water quality should be okay. Sometimes they are graded. They are in high densities, and it is recirculated water. So when there is something in the water, it comes back in the basins every time. Although there is, of course, a biofilter to get rid of many of the, well, perhaps kind of possibly problematic factors in the water quality. The elvers become yellow eel in the farm, and yellow eel, they are at higher temperature. There is a fast replication of at least parasites and bacteria, and some viruses also like high temperature. So then these yellow eel may be used for restocking, but here is the point. The eel farmers ask for a kind of testing before they restock the eels. So then we get it at the laboratory, and when these eels show clinics, they are already not accepted to restock. That's the first thing. And also we test for certain viruses, for Anguillid herpes virus 1, for instance, and for absence of the swim bladder nematode. Of course, it is depending on what is already in the open water where you restock the eel. So for instance, it's mostly yellow eel for restocking or elvers, but also silver eel sometimes, but that's already very late phase. So it is really the question. You have to compare the origin of the eels which you are restocking. They should be clinically healthy, and you should test for certain things before you restock. So the quality important, are they clinically healthy, and check for, well, appropriate parasites, bacteria, and viruses. So I come to my conclusions. Actually, there are various eel pathogens in Europe, partly introduced by imports, viruses, bacteria, and parasites. So I've already told the Anguillid herpes virus 1 was introduced. Bacteria, well, it is at least we could say that the Pseudomonas angliseptica is an interesting one, which is also causing disease in sea bream and sea bass along the French coast, where the elvers actually pass by the cage culture of these cultured fish. So perhaps there they acquire it. We don't know. Parasites, yes, that the Angeliculoides is not originally from Europe. So it was introduced via introduction of elvers or other stages of eels. So some of these disease or parasites may cause severe eel disease and mortality, more when eel is stressed. With restocking, more interaction, wild and farmed eels, so two ways. So from elvers to the farm, and later on for restocking. Any introduction of populations implies a disease transfer risk, restocking too. However, many pathogens are already present in the wild, and therefore I believe in targeted monitoring of restocking eels to add to the wild eel population. So compare what you have and compare where you go to. So a long-term effect on the recruitment is actually not known to me yet. And I think we really need monitoring to investigate this. And this means also sacrificing eels again. Do we want this? What should we do? Well, to my opinion, we should look for an optimal environment of for the eels. We restock eels regionally and not go to the other side of the world to restock. I think that's really a risk, and we have seen this with the introduction of some pathogens and quality restocking. So targeted monitoring of restocking eels. This is a former eel workshop, which was really nice with all eel-minded people. And again, we are in an eel-minded group, so I'm really happy about this. There have been many people working on the eel disease, so here they are mentioned, and this is our PhD student who had an excellent job, did an excellent job, and so actually I would like to thank you for your attention, and yeah, I hope this kind of gives an idea. More info can be found on our website, on the page for fish diseases of BBVR. Thank you.
Thank you very much, Olga. That was a very thorough going through, I think, of eel potential diseases to eel and their origin and potential effects. I certainly didn't know all of this beforehand. Anyway, I will swiftly move on to Isabel Domingos, who is an assistant professor at the Faculty of Sciences at University of Lisbon, and also researcher at the Marine and Environmental Sciences Center in Portugal. She has been involved in many projects and much research on European eel, and also provides scientific advice to the Portuguese administration. And she's a participant in the ISIS-IFAC GFCM working group on eel since 2008. And she's also involved in national application of the CITES Convention in Portugal. But she will talk now about one of the few studies actually that we managed to find, which looks a little bit more on eel and ecology, you know, and the effect of eel on the wider biological community. Welcome, Isabel.
Thank you, Niki. Good afternoon to everybody. Just a second, I need to, there's something wrong in here. Sorry. Is it the full screen? Yes.
Yes, indeed.
Okay, thank you. Thank you. Sorry, there was a problem in here. So I'm going to talk about an experiment we did on restocking and the effect that it had on the biological communities. But before that, I would like to present some information that I think is important for us to understand how restocking can have, sorry. I am very sorry. So picking it up, sorry again. So I'm going to talk about an aspect that I think it is important for us to understand how restocking can work better or worse, apart from all the topics that have been addressed by previous speakers. So the first thing I would like to focus is the fact that we all know that glass eels come from the sea, and they come into continental waters, and they tend to colonize river basins from the river mouth up to the upland areas. And they proceed this colonization until a certain period when they find the perfect or the good habitat conditions. Although we know that they can shift between brackish water and freshwater habitats throughout their growing period. But the question is, what are the good habitat conditions, and what are we doing when we somehow restock certain river aquatic systems with eel? And picking up the regulation, I would like to focus 3 aspects that have been kind of the driver of many of the restocking that has been done lately. And the first thing was, and it was written in the regulation somehow, member states that had a glass eel fishery should reserve 60% of catches for restocking in their country or elsewhere. And this would feed the restocking needs from other member states, and to guarantee 40% silver escapement. Member states have been restocking, some member states, and in some cases, or quite often, when glass eels or elvers are kind of trapped below obstacles, there is quite often the action of putting them in transferring them to other river basins or to other to tributaries where mortality should be highly reduced. But in any circumstance, or in most circumstances, carrying capacity and habitat quality are not taken into consideration, and they can end up in mortality that is seldom not quantified or hard to quantify. And I have listed in here 2 aspects when I talk about carrying capacity, although there are numbers, kind of biblical numbers, and I am exaggerating when I say this, but it's what I feel in some circumstances. And the number, but it's not only the number, it's the size of the eels versus the weight of the eels that I'm going to put in a specific aquatic system, and also the habitat quality. And I'm not talking about contamination or parasites or whatever. I'm talking about food protection, and also another threat that is seldom addressed in here, which refers to exotic species. All of these increase the mortality. But picking up the subject that brought me here, I'm going to talk about an experimental restocking action that we did in a tributary from the Mondego River Basin. The tributary is the river Seira, and this tributary is probably one of the most fragmented tributaries of this Mondego River Basin. And the first thing that kind of shows up to your mind is, why on earth did we choose a tributary that is so fragmented? Because this was an experiment to see the results of the action. This is the study area, the extension. It goes for 27.5 kilometers. And we have kind of designed this experiment focusing on the obstacles up and down. If we want to compare the effect, if we want to interpret the results of a stocking action, we need to do the stocking, obviously, which we did in 3 sites that are marked with this green arrow, site 1, 2, and 3. And but we have also considered 2 control stations, because we wanted to know or to know if what happened in here was a result of the restocking or was a result of natural variation. And we had to kind of exclude that possibility, and therefore we created 2 control stations, let's say like that. So 3 sites were restocked, and also you see this square, red square, where 2 obstacles are marked, and these obstacles are they are unsurpassable, which means that what we have stocked in here will somehow remain in here. This is the only way we could control something, because when you stock on a river, on a lake, you have a closed system. If you do a stocking on a coastal lagoon, you may have a closed system, but in here, a river is never a closed system. So it would be very hard to study or to analyze what was going on. So these downstream and upstream control stations have been marked, and we compared non-restocked versus restocked sites. Obviously, we did this, we wanted to answer 2 questions. One of them would be, how did the biological communities in the system react to this restocking action? And how did the population, the eel population, the one that we have restocked, how did it react? So we had to create a pre-assessment and a post-assessment that we have addressed throughout this study, which was a short study. So the study started in April 2014 and ended in April 2016. There are 2 papers that, after all, resume what I'm going to present in here. And this is the situation, or how we did it. We have, in previous to restocking, we have done the sampling to create the reference situation condition, to know the reference conditions. That is to say, how were the fish community, how was the fish community in there before restocking, and how was the macroinvertebrate community in there before restocking. So we did the first sampling at the beginning of April, and then in the same year, the same month, we did our restocking actions, and we have monitored it for quite for some while. So the restocking occurred in 3 sampling sites, 1, 2, and 3, and the number of glass eels that were released on each site were the ones that are marked in here. In total, we have released 18 kilograms of glass eels, which corresponds to 60,000 individuals, with a density of 0.9 individuals per square meter. In April 2000, we did this in April, and then we monitored it quarterly, and in April 2016, we went there again. So the basic procedures for this experiment are the ones that are presented in here. So we had to focus on 4 aspects. One of them was the species itself, what happened to the eel, and then how did the other aspects move through, change throughout the study period. So we had collected environmental parameters, the fish community, and the macroinvertebrate community. We did this on a quarterly basis from April onwards. And the feeding impact, that is the macroinvertebrate community, we did it on a yearly basis, a pre-assessment in April 2014 and a pre-assessment in April 2015. So the information collected throughout the period was the one listed below. I'm not going to list it, to say it, but for the eel, it is obvious. It was how it grew and the density it had after the restocking. And for the fish community, we have been doing all the sampling and estimate the density. The same applied to the macroinvertebrate community. So this was what we did the first year. This is the practical work. The fish species community, we had to go there with electric fishing, identifying, quantifying, measuring all the the fish that we caught. Then we used kick sampling to analyze the macroinvertebrate community, and we also collected at each sampling occasion the environmental parameters, namely depth, type, the substrate, etc. And the results of this work for the macroinvertebrates, in fact, there were differences in the macroinvertebrate community from after 1 year of restocking. And these changes, so the changes occurred exactly, the differences were exactly where we had been restocking. So this is significant for restocking places. The control station, the one that we restocked, and another one that we restocked. And this might obviously be related, because we can't forget we had the control stations to discriminate between what was natural and what could have resulted from the restocking action. So we anticipate this could only derive from the restocking action. And looking at the macroinvertebrate community in 2014 and 2015, there weren't that many changes. Somehow there was a decline in sensitive species, the ones marked with yes, contrary to the tolerant species that didn't decline. So the decline was obviously in the sensitive species and not in the other ones. There was also a change in the type of macroinvertebrates, that is to say, there was a change in those, or the abundance of those that had a burrowing behavior and those that are bottom dwellers. So somehow there was a change in the abundance, or in the, yes, relative abundance of these two types of macroinvertebrates. However, when we look at the differences, we look at the number of individuals, macroinvertebrates, the diversity, the species richness, and we see that somehow there seems to be an increase in the number of, in the second year, which somehow doesn't make that much sense, or it's unexpected. But looking at the control stations, it is clear that there is a difference in the sites that have been restocked, contrary to the ones that were the control stations. So this is more than evident, and but still, I would like to highlight that we used an integrity or biotic integrity index, this one that we used for the Water Framework Directive, and the macroinvertebrate community didn't get worse. On the contrary, it improved, as you can see in here, in 2015. But this is also an unexpected result. As for the fish community, there were no changes, no differences between 2014 and 2015. We can think of many causes for this. One of them is the fact that the glacials were somehow small, and therefore the time that has evolved since restocking might not have been enough for us to see any competition. And also the mouth gap between the glacials, or the elvers, and the other fish species, which are mainly cyprinids, and most of them, they are very small, except for the barbel, which is the largest species, but the other ones are small, so we wouldn't expect that much competition between these species. But it's only 1 year, and therefore we can't take this, we have to be cautious about this. Looking now at the eel density, how it evolved, and how the growth has, how it has grown, we look at this graphic, the first one. So we have site 1, 2, 3, and 4. This one was a site that I didn't focus it, that where we didn't release any glacials, but since there was not an obstacle between site 3 and 4, we would expect the animals to disperse, and that is what we somehow see in here. So, and also another thing, that aspect that I would like to highlight is the fact that there seems to, these are obstacles. There was an obstacle between site 1 and 2 and 2 and 3. So there seems to be a concentration of or a higher density because of the presence of obstacles. So the densities varied depending on the sites. They are higher downstream of obstacles. And this wide dispersion I would like to highlight, because it means that naturally those animals could progress upstream. There was a dispersion of the animals released of around 4 kilometers upstream, but there were some zero catches, which we have difficulties in interpreting because they might be, the first thing is, was mortality increased because of this? Or is it because they dispersed? Or also, because if we look at here, January is the coldest month, and it seems that during winter, there is a decline in the number of animals that have been electric fished. As for growth, there were differences in the growth rate between sampling sites, between releasing sites. Site 1 is the one that is more successful, or more not successful, where growth was more successful. And but still, in 1 year or 2 years, because in here we included the 3rd year, so it was the one that was not done quarterly but yearly, we have got growth rates varying from 29 millimeters to 59 millimeters per year in the 2 years. And these differences might be related to the variability that exists in eel growth, or also to the local conditions, and eventually and possibly the food availability on each site. When we look at these results, we get somehow doubtful about many aspects. Before going to there again, I would like to highlight that this graphic in here is the one that has been produced in the DIC's workshop on eel stocking in 2016, where we see in green all the countries that have restocking. In here there's no information, and in here there is no restocking. So in general, what we see is that within European Union, there is a high demand or a high need for restocking. And most countries rely on it to, Reinhold mentioned the fisheries, but we can also think that, he also mentioned it, that sometimes some of the restocking has a positive effect, and it might increase the escapement of silver eels. What do I mean by there's a hard work to do? There's a hard work to do because when we look at this map, we realize that some countries don't have contact with the sea, that the migration or the escapement is hard to understand under the circumstances, although some international rivers would account for it. But also the fact that when we look at all this diversity of habitats, we think that they might all occur in this area. And in fact, some conditions are not the best for the eel or for restocking. I have just deleted these ones because, and Reinhold also mentioned it, this is a dam, and restocking above dams is counterproductive. It shouldn't be done. The same applies to rivers that are regulated and have nothing but sand in the bottom, and where the habitat available is not favorable for the eel. So the only eels that exist in here are the largest ones that are kind of ambushing some prey, some fish. But there's a big difference in size of eels throughout the riverland, and some conditions are more favorable. For instance, in here, we have a huge diversity of habitat where you can find not only a higher abundance of eels, but also a diversity of sizes, because there are lentic areas in the bank sides and lotic areas in the main course, apart from the existence of a high abundance of prey, because many of them are in the stones that exist in here or in the boulders, and most of them in the bank sides are in the margins, in the bank sides. So with the vegetation, macrophytes, etc., this is the perfect habitat for certain eels. I've also chosen to put a lake in here. Now I've chosen this lake because it looks somehow, it looks attractive for restocking, in the sense that the bank sides may not be that deep, and there is a high primary production, which is the basis for the production of food. A rich macroinvertebrate community can exist in these areas, contrary to this one, where you have just the regulated river with some trees on the bank sides. So in the end, I would like to highlight that this was 1 year study. So the results that are in here, that are presented in here, are somehow, should be looked upon with some caution. And based on this, I think it is important that we, whenever a restocking action is done, we first of all identify what is a suitable habitat for restocking, assess the carrying capacity of the donor recipient, which is something that is somehow not done when, or not based on evidence, is somehow, as I said before, a biblical aspect. And also estimate mortality, because from our results, we don't know, we are not sure if the low or if there was a dispersion of individuals along the river course, or if there was really some mortality. And there is no way we can somehow know that. So looking at, I forgot to mention something important. This action was done within the framework of a project that aimed at reestablishing longitudinal connectivity within the river basin below hydroelectric power, because hydroelectric power is a problem and it will not be easily solved. It contributes to increase mortality, and it is not good habitat for restocking, not only because of mortality when silver eels are going to escape, but also because food or the prey are not that abundant, especially because those are deep areas. The weir that exists above the dam is usually extremely deep and not the perfect habitat for eels. Only large eels that have to prey upon fish, but those are very, at least in Portugal, those are very rare compared to the males that are the dominant sex in our river basins.
Isabel, Niki here. Are you
Sorry.
finished? Yes, because
Yes.
yeah, we're very much running out of time, and we were going to have a small panel with you.
I'm sorry.
Reinhold and Argyrios at the end, answering some prepared questions. So can we shift from the presentation mood to the panel mood for the last 15 or 12 minutes or whatever we have? Is that all right? So I wanted to ask all of you, and thank you very much to all of the speakers for your presentations today. Considering all of the different aspects that we've been covering today and the risks and uncertainties involved, and also the ICES advice, obviously, which sets the precautionary principle, we should have no fishing of eel on any life stage. Do you think, and I'm directing this at all of you, but we can start with Reinhold maybe, is there currently any scope for using restocking as a conservation measure to support the recovery of the European eel population?
Well, I would not make a final decision on this. I mean, there might be instances, and I think Miran brought this up in the chat, where it is shown, and also Isabel mentioned that when it is shown to a certain donor river or donor estuary, that it is proven that there is a certain number of, that there is a surplus of glass eels to arrive in a certain estuary. Given also that some of the donor rivers are also, for sure, not native at all. They are also heavily anthropogenically influenced or impacted in some way. Then there might be good arguments to fish a certain amount of glass eels and to restock them elsewhere. But what we saw also from Caroline's presentation, I think it is sure that at first these should be recipient waters that should be very close to where the donor waters are. So it's more about probably assisted migration or to search for high quality waters nearby that could really, where also scientifically there could be good arguments to take a share of the natural glass eel arrival and transport it nearby. But these are, from my point of view, would be rare instances where for very good reason this concept could be broken. But overall, restocking as it is practiced now, like a commercialized measure that has no scientific background on a large scale, that I think this must be really changed. There is a need to realize from a management point of view, especially going back also to the eel regulation, that it can never be that a regulation or a management framework actually builds a legal obligation almost for restocking, as long as there is no scientific proven evidence for the benefit of restocking for the overall stock.
Yeah, I thought I should maybe clarify that I'm really asking now from the population point of view, and not, you know, as a matter of fulfilling the 40% escapement target. Thank you. So Isabel, do you want to go ahead and answer that question as well? Do you want me to repeat the question? You're muted, I'm sorry.
No, you don't need to repeat the question. Yes, it is true that I agree with we can't say we shouldn't do restocking, but we should really look at restocking not as a commercial business somehow, or at least it needs to be monitored. It's no use for me to restock with a certain amount of glass eels or elvers that I have estimated to be the ones producing the target I need to get. It needs to be assessed, it needs to be monitored, and it needs to really to see if it is working, because it might not be working.
So, and Argyrios, I don't know why, with your presentation, obviously, so the Mediterranean has a completely different history in a sense than the other regions. You don't really restock in the same way, even though you mentioned that Greece and Italy has started restocking sort of longer distances recently, but you have more like a translocation from outside lagoons into lagoons. I mean, what is you were quite clear about the GFCM conclusions, I suppose, but what's your personal reflections on this?
Well, to tell the truth, from the point that we don't have any specific evaluation of these measures' effectiveness, we cannot say that stocking is a conservation measure that can assist in the recovery of the stock, since, as we said, we import glass eels from UK and France. So just translocating the species is not, I think, a solution to this point. And as you have seen in the table with the scenarios, there are measures that are more effective than restocking. Restocking was, let's say, the easy solution for the preparation of the management plans in order to achieve the 40% of escapement. So you have a measure, you import glass eels from another place, you stock places that probably it's not the most appropriate eel habitat, and then you monitor the escapement. But okay, it's not the recovery of the local stock, it's the eels that you import from another country. So for me, it's not the best solution. We have to find measures that will assist the local population. Yes, I cannot suggest the total closure of the fisheries, because it's the whole socioeconomic situation behind the fisheries. So a partial closure of the fisheries, or minimizing the fishing efforts for specific fishing gear, is a way to increase the amount of spawners that are managing to go to the sea and then start the migration towards the Sargasso Sea, and then just adding more and more seed over there. And we don't know also the mortality of these actions. It's increased, probably it's increased, because we have a lot of different stages during the catch of the glass eels to the restocking. So for me, it's not the best solution, and we need to find a way to further fill the gap of knowledge in the Mediterranean. The GFCM project was a start because we managed to find data that nobody had an idea that exists, and we start filling the gap, and we need more work to be done.
Okay, thank you. That was very helpful answer. I'm going to try to squeeze in a couple of more things here. So Reinhold, you were talking, I mean, all of you in some ways have, well, maybe not Olga and Caroline, but you've been touching on obviously the issue of hydropower plants and other migration obstacles and so forth. And you mentioned that restocking, particularly in the Baltic region, is actually contributing to increasing mortality in hydropower plants because there's quite an extensive restocking above them. Do you have any idea how large this sort of artificially created mortality is, where actually rather than contributing to recruitment, we're essentially sending the eels to a certain death?
To be honest, we are really lacking data on this. So most of the countries do not specify where they actually stock their juvenile eels. But there was a large discussion now also in Boldfish about potential measures, and it is clear that some countries really are kind of fearful of stopping any restocking upstream of obstacles, because then they would not be able to restock most of their waters. This is, of course, a very difficult argument. We know also from Germany, for example, that really eels are stocked far upstream and upstream of chains of hydropower plants, like Lake Constance, for example, is a good example. It is really far for eels to reach at any aspect the Sargasso Sea, because there are several hydropower dams in between, and turbine mortality on average is about 40%. So it is very clear that this kind of restocking has nothing to do actually with the recovery of the stock. So it is clear that if any restocking measures should be continued, then we really need a prioritization in the different countries about rivers or other water bodies that have free access to the sea, and also where other like parameters like pollution and also fisheries impacts are really low. And this is an exercise I think that should be or needs to be done all over Europe. Imagine just if the number of recruits is further declining, which is not at all excluded, then at a certain point we will be short of glass eels, and then we will be in need of decisions if we really want, or if managers want to continue with restocking, to choose rivers where the chance for an individual eel to ever reach the Sargasso Sea is really high.
Okay, I'm going to go because we've got almost no time left, but I'm just going to try to squeeze in, and please try to give me a very very short answer. So oh, never mind. Anyway, Isabel, I wanted to ask you one last question. You know, because your study, even though it was just a very short time-limited study, showed some positive effects on biodiversity. And obviously what we've seen over the last 100 years is that the eels have used to be one of the most common fish species in almost all European waters, and it's disappeared. So it's obviously, by disappearing, has had an effect on ecological communities. And I personally, being so engaged in the eel, I wish there was much more studies on the sort of the ecological side of eels and the effect of you know, it disappearing and then the possible reintroduction. But I just wanted to ask, when you looked at this riverbed, I mean, if you imagine that you would be able to remove some downstream barriers, so basically expanding the habitat of eel, I assume, well, or what do you think, would we see the same sort of positive effects on the benthic fauna as you did in your study, or?
It would depend on how degraded or affected the system would be, because after 30 years of having no eels in there, which was the case, the system had just got into a new equilibrium, and therefore the relation and the trophic relationships among all species, benthon, macroinvertebrate and fish, have kind of restructured, and therefore the eel was a new incomer in there, and therefore it affected it. Maybe because, and I think the result was like that, because the eel is very much ubiquitous and plastic in diet. Therefore, she will always eat what's more available at each moment. So she's not very focused on a specific item, and that will contribute to kind of cut the grass and let it grow, if I can use this symbol, in the sense that it would never explore a unique resource. It would feed on many, and this would leave space for all the trophic relationships among the species that are in there. But this needs time, because she is a newcomer in there, and therefore, if I just solve the connectivity, there are many things to change, because the system is completely different. Sometimes it's deeper, etc. The habitat and the bank sites are completely destroyed, so the food is less available. It depends. It depends. It will always take time. That is my final conclusion. It will take time until all the community gets adjusted again.
So are you, in a sense, saying that even if we were to refocus, several of you have actually said that restocking is not a very effective measure for conservation of eel. It'd be much better if we didn't distract ourselves with restocking, but we spent more time and money and effort on the other measures that would have more longer term effects. But are you saying then, Isabel, that dam removal or obstacle removal, which would be one such measure that we know has also been much less implemented in the national eel management plans, that would also need to take some quite careful consideration, and you would have to pick your sites carefully to get the maximum effect?
Yes, and another thing I would like to highlight, because I consider it important, we are focused on the longitudinal connectivity, but we forget the laterality of aquatic systems. And by saying this, I mean all the salt marshes, the marshlands that have been destroyed by land reclamation. They were perfect habitat for glass eels and delvers to set before proceeding upriver. And that is what we should focus our attention in the near future, to recover those habitats.
The ones that we need to reflood to save us from the effects of climate change.
Exactly, those ones. So this is a conflict of interest. Yes.
Okay, yeah, so I'm sorry, we've completely run out of time. I was hoping that we'd be able to have a longer panel debate, and maybe next time I will need to be harsher with giving time limits to the speakers. But I'm really grateful for your participation, all of you today. It's been great to listen to all of this knowledge, and I've noted that you've also been answering questions in the chat as we've been going along, which is very helpful. Thank you so much for that. And then I just wanted to flag that we have a sort of an informal eel coordination group in the NGO community, and it's obviously a very important and ongoing issue this year. So we've essentially agreed that we will organize a series of webinars on eel. This is just the first one. So I'm hoping to speakers and all of the participants that you will be able to join us again for future webinars over the months to come. Thank you so much, everyone. And to all of the speakers, very heartfelt thanks. And thank you also to the FishSec staff that supported with all of the practicalities in preparation and during the meeting. Thank you so much.